So talking about the next gram-positive bacillus, and that is Mycobacteria. I hope you guys are going in continuity and studying about the gram-positive bacilli first. So under this, we are first going to start off with what is the structure and what are the types of Mycobacteria that you need to know. Of course, we know the famous Mycobacterium tuberculosis. Then we have the non-tubercle Mycobacteria, and we have Mycobacteria leprae, that is leprosy. So let's get going and start off with first what is Mycobacterium TB, which is the most important in our country. And there are some things that all of us know so well about TB that I don't have to write it down for you. It's going to come so organically to all of us, isn't it? So the first thing that we have is that it is going to spread by droplet nuclei, that is by close contact, and it can spread through ingestion also, like you read about unpasteurized milk. All of you have read about intestinal TB, right? So ingestion is also one of the roots of it. We know that. So what is the main thing that we have to know? That is the structure of tuberculosis. So if you read this MTB wala structure, there are some things which you need to focus on. Of course, it has a cell membrane like any organism. Of course, it has peptidoglycan because it is gram positive. It has something known as arabinogalactin. So I can see something Ag. What is it? Ag is going to be arabinogalactin. That is something which is important in its surface. Also, I want you all to know there is something similar known as lipoarabinomannan. I'm just enumerating them. Apart from that, there is something called mycolic acid. So what are the structures and their functions that I have to know? So first I told you mycolic acid. Mycolic acid is something acid. It is responsible for the acid fastness. Haven't I told you that MTB is acid fast? Hai na? So mycolic acid is responsible for that. Then there is something called cod factor. Remember, cod factor is responsible for the virulence of the organism. It's one of the virulence factors that this organism has. Next, we have something known as LAM. LAM stands for lipoarabinomannan. LAM is going to be lipoarabinomannan, and please note that this is responsible for a couple of things. Firstly, it is responsible for the very, very important part, that is how the TB attaches to our cells. Second, it is responsible. So have you ever wondered that whenever Mycobacterium will come inside our body, there will always be one cell which is ready to eat it up, which is that hungry cell always ready to eat it up? Macrophage. So if macrophage has eaten it up, phagocytosis should happen, MTB should be killed. This means I'm having MTB, this means macrophage is not able to do phagocytosis. Why? Because lipoarabinomannan is going to inhibit the phagolysosome formation. There is a problem in phagocytosis that it causes. So remember, it inhibits the phagolysosome formation. The patient doesn't have the ability to kill that MTB. And thirdly, which I will tell you in a while, there is now something known as a lipoarabinomannan urine assay. It is actually used as a test in urine, and this test helps you in diagnosing particularly TB in HIV positive individuals. So I'll be coming to that also in a while. I think we've understood the virulence and the antigenic properties. Now let's go on to the clinical features. I think everyone knows one thing, that we have something called, I said, some things will come so easily. One is something known as pulmonary TB, and then there is extrapulmonary EPTB, right? So pulmonary and extrapulmonary, my first concern is pulmonary tuberculosis. So when I'm talking about pulmonary tuberculosis, please note it can be primary, secondary or disseminated. For primary, I will say the first time that the infection occurs. What is the location going to be? It is usually subpleural in location, point number one. And secondly, this is going to occur where? So it's going to be like a tongue twister. Dekho, yahan par hota hai. If I can highlight it, it's going to be somewhere here. Means, what is the tongue twister? It's going to happen in the lower part of the upper lobe and the upper part of the lower lobe. So what did we say? It happens in the lower part of the upper lobe and it occurs in the upper part of the lower lobe. So this is a kind of a tongue twister form, but this is the location where it's going to occur. That is primary TB. Now when we talk about secondary TB, which is basically a kind of a reactivation TB that occurs, if we look at this, we will realize that this tends to occur at the apex. This occurs at the apex, and this is also going to have a cavity. Now, this means radiologist is going to tell me a cavitatory lesion that is present over the apex, isn't it? But I want you guys to make a list. I know cavitatory lesions in lungs are like way too many, but main bata do, on which you get a question. So when they say that in the lung there is a cavity that is present, of course in our country you will have to think of secondary TB. And TB has a very famous fungal infection which resembles that TB. Which fungal infection resembles just like TB? Histoplasmosis. Histoplasma also resembles very clinically a lot like TB. Apart from that, there is one disease called bronchiectasis that also has cavitatory lesions, and which is the cancer that can come to us as cavitatory lesions? Squamous cell cancer. I'm not saying these are the only cavitatory lesions, but 99% you're going to get a question on this. Secondary TB, histoplasma, bronchiectasis, as well as the squamous cell carcinoma. Now, before I go on to the other part, I want to tell all of you this. I want to say TB is leading to a cavity. Because the person had TB, because of that he ended up having a cavity. Can you tell me on the other hand, if I ask you that if there is already a cavity present, if there is a pre-existing cavity that is present in the lung, which is the fungus that is going to come and grow inside it? Opposite. Cavity is already there, fungus will just come and sit inside it, and that is known as aspergillosis, and then that is known as aspergilloma. So look at the two differences that you have. TB is leading to the cavity. TB is causing the cavity, whereas aspergillus is it's like an opportunist. It says, acha, there is a cavity over there, let me go and sit inside it. So listen to these two questions very carefully. Which fungus can cause the cavity? Histoplasma. Which fungus will go and sit in a cavity? Aspergillosis. Let's move forward. You are done with primary, secondary. Of course, you are left with the disseminated part. Disseminated part mainly means that it has gone to different parts of the body, and that it has gone because of hematogenous spread, via the blood spread it has gone, and when it goes in different parts of the body, it results in miliary TB. So for example, like it's gone in the liver. So miliary means it's going to be like this, like dot dot. So miliary means like the millet seeds. That is how it has got its word. Millet seeds are chota grains, na? So like this dot dot dot, it has come up as miliary TB. So now this means when I'm talking of so many other organs, I can easily talk about after pulmonary TB, what are the extra pulmonary TB that are going to be there. And I hope you remember the most common extra pulmonary TB is going to be the lymph node tuberculosis. So in our country, most of the patients, either they come to you with lung TB or they come to you with a lymph node over here. So that means it's a lymph node TB. But there are other organs also, like in the kidney. And if you go to your pathology lectures, I have taught you that in the kidney there is TB is known as putty kidney. In urinary bladder, if TB occurs, then you call it a thimble bladder. You call it a thimble bladder. If it occurs in the skeleton, and I'm not talking about the entire skeleton, I'm talking about the spine. What is that known as? The pot spine. So these are some fancy words, and we all know examiner loves fancy stuff. When we are talking about CNS, yes, this can go in the brain as well. Can it go into the skin? Of course. When there is TB, when there is primary TB occurring in the skin, we call it lupus. We call it lupus vulgaris. That is when primary TB is occurring in the skin. Whereas what do you understand by scrofuloderma? So what is scrofuloderma? Derma I've understood is to do with the skin, but scrofulo tells me it is spreading. So this means if this is the skin, like for example, you have the skin of the supraclavicular region, but this means there is a TB below in the lymph node. That TB is spreading over here. So scrofuloderma basically means there is spread to the skin from an underlying focus. It's not actually of the skin, but from an underlying TB focus, it has come onto the skin. So remember, lupus vulgaris is going to be in the skin, whereas scrofuloderma spreads from below to the skin. These are some examples. Otherwise, you know, TB can happen in possibly every organ of the body. So now out of these, you need to know what are the different foci. These are very fancy names they ask you in the paper. So what I'll do, I'll draw one lung over here, right? If TB occurs in the lung like this, TB yahan par hua, you will call it gone focus. What is gone focus? When TB is involving the lungs, you call it the gone focus. Focus ke baad you will convert it into a complex. What is a complex? You will say, ma'am, if the lymph node also gets involved, if both of them end up getting involved, now you will call it a complex. So this means there is going to be involvement of lung plus the hilar lymph nodes. That becomes a gone complex. And if this complex gets K for K, if the radiologist says that there is calcification that is seen in this complex, you will now call it Ranke's complex. So repeating, if there is one foci likha hai, to one focus, one site, and that is going to be the lungs. If complex word is written, that will mean that there are two things involved. There is lung and lymph node. And if calcification word is written, complex in terms of Ranke, K wala complex is written, it is calcification. Okay. Now let's move on to apex supraclavicular inflammation. infraclavicular? For that, I will have to draw one more lung in front of you. Look at this and tell me one thing. If you look at the lung, which is that beauty bone that crosses the lung? The clavicle. So we all know that the clavicle is going to cross the lung just near the tip it crosses. No? So now, if I say what are the parts of a lung, you will say the first part is over here. Tip par kya hai? You will say that's the apex. Then you will say, okay, the little bit area left is supraclavicular part and rest everything that is left is infraclavicular part. Do you agree? Top par there is apex, then there is a supraclavicular, then there is an infraclavicular, and now you will remember in that order you will remember the mnemonic SPA. So what is SPA? SPA means on the apex we have something known as Simon focus. On supraclavicular area we have the Pearl's focus. And in infraclavicular area, we have the Assman focus. Okay, everyone, in the exam, first you'll think of the order. You will say, okay, top par there is apex, then supraclavicular, then infraclavicular. So that is Simon focus, Pearl focus, Assman focus. So you've clearly made a mnemonic SPA over here. Now let's come to other organs. We are too much into lungs. Let's go to liver. Liver mein what is going to be there? It is referred to as the famous Simon focus. Don't get confused. Which is heavier, out of the lung and out of the liver, because lung ka apex was Simon and liver which is heavier is Simon. So heavier name I have got, bigger name. If it occurs in the vessel, V for vessel, V for Vigot. So I have a different spelling, W. So Vigot's focus is basically going to be TB in the blood vessel. So simple, Vigot for vessel. And brain mein now think guys, brainy people are always rich. Okay, so Rick's focus. Rick's focus is going to be in the brain. These are all the foci that you have in tuberculosis. Now asking you about the hosts. This is just to quickly read. It's just not humans, but also cattle, dogs, cats, animals can also be hosts. I just wanted to give you that one line of information and that is sorted. So humans and animals can be hosts. Now I think I can come to you with the diagnostic ability. So in the diagnosis, guys, I'm not taking up what you already know. I'm not going to tell you that TB is going to show you cough with sputum, with hemoptysis, with weight loss. I mean, no one's going to ask you that. And even if you ask a non-medico walking on the road to just tell me what are the features of TB, everyone's going to tell you. Let's not just waste any time on it. Let's go on to what the work of a doctor is. That is a diagnosis. So let's assume I'm talking about a pulmonary TB. Let's assume I'm talking about a pulmonary TB. So what is the specimen that you will get? The specimen that you will get is going to be a sputum sample, of course. So how many sputum samples should you be taking? You have to take 2 sputum samples. Means the first one is going to be a spot sample. The day the patient has come to you, over there only tell him, come give me 1 sample right now. And then tell him that you have to also give me 1 early morning sample the next day. So 1 spot sample and 1 early morning sample has to be given. Also, please note that you should have at least 2 to 5 ml of the sputum sample, but it should be sputum. It should not have blood, and especially you have to consider it should not have saliva, because in our country, people just tend to, in that container, they spit out some saliva and give it to you. So prior to sample collection, that counseling is very, very important, that you have to give me a sputum. You have to cough out the sputum. It shouldn't have blood. It shouldn't have saliva. And you have to give me 1 sample right now. 1 sample you will be giving me tomorrow morning. That is the counseling. But if the patient says, by chance, the patient says, I have dry cough. Earlier I was having sputum, that now these days I'm not having any sputum production. It's dry cough. I waited 1 or 2 days. It's dry cough. I'm not able to give you the sample. Then what are the other options you have? You'll have to do some techniques. You will have to take laryngeal aspirates, bronchial secretions, bronchoalveolar lavage. Then you find out how to take out the secretions from the lungs. What if it's a child? Now please note, in children, we might have to do a gastric lavage. So how gastric am I talking about? Because what happens, that especially I'm talking about children. Children don't tend to spit out the sputum. Whenever they cough, they tend to swallow their sputum, right? Because young children don't know that they have to spit out the sputum. So they might end up swallowing the sputum. So that is going to be, now this means all that sputum is going in their stomach. So now you have to do a gastric lavage. So please note, for gastric lavage, you have to always consider that we should take an early morning gastric lavage, and within 4 hours of collection, the testing should be done. within 4 hours of collection, the testing for TB should be done, because anyway gastric lavage is very, very acidic condition and you don't want your TB to die off. So simple, that only if it's a child who's swallowing a sputum and you're not able to do bowel and other things, you do gastric lavage. Okay, so this is all that I considered. You were assuming that this is a case of pulmonary TB, but it's not going to happen all the times like that. What if it is a case of extrapulmonary TB? And the most common situation I told you was lymph node FNAC. What are you going to do? Lymph node, to FNAC karoge, FNAC mein pus milega. This is the most common thing. This is what we do. 90% of our FNACs in our country are going to be this. People coming to you with lymph nodes and you put needle into it. You know, on radiology, there was pleural effusion. So then you'll go in for a pleural tap. What if this is a case of meningitis? So how does the TB ka CSF sample look like? That's a picture in front of you. You can see a cobweb coagulum formed. Please remember this question for your Viva as well as for your MCQs. Cobweb coagulum is seen in which particular condition? It is seen in tuberculosis. Very, very important. What if it is a kidney problem? So you will take urine sample, but you know, one urine sample is never enough. You have to take 2 to 3 days ka urine sample. You have to centrifuge it. Then you might be able to isolate TB. So isolation of TB from urine is not that simple. So basically nothing for you to learn. Only thing is, depending on the site, you will take the sample. Lymph node hai to FNAC, meningitis hai to CSF, kidney problem hai to urine. The only question you can get out of this is meningitis mein CSF of TB shows you cobweb coagulum. Now let's move on to the fact that extrapulmonary TB to theek hai. If for example, let me come back to the usual. Let me say there is a pulmonary tuberculosis ka case, and from this pulmonary tuberculosis patient, I have suspected pulmonary TB patient. I have got a sputum sample. So what are the things that I'm going to do? I know you're waiting for that ZN stain. I'm coming to that. But you might have to concentrate that sputum sample by a method known as Petrov's method. And Petrov's method just means that you're going to take the sputum and you're going to add 4% NaOH. You're going to take the sputum and add 4% NaOH. But nowadays, there's a further modification done, and that is NaLC and 2% NaOH is added. That is what we are using nowadays, NaLC, that is N-acetyl L-cysteine. N-acetyl L-cysteine, that is what we are doing nowadays, and that is what is going to be the current concentration. So this is mainly because you'll say, why do you want to concentrate? Because I told you, sometimes the sputum will not have so many bacteria only. You won't be able to pick it up. You want to increase the load. So you will concentrate the specimen. So collection is done, concentration is done. What is the microscopy that you are ready for? What is the stain that you're going to do? The Ziehl-Neelsen stain. The Ziehl-Neelsen stain. I know it's a part of general micro. Can you quickly within a minute revise the protocol of Ziehl-Neelsen stain? Remember, I gave you a mnemonic. Class has asked mnemonic. Yaad aa raha hai? I'll write that once again for you, because this is the day that I taught you that for. Class has asked mnemonic, which basically means that there is going to be carbonyl fustian, the first thing that is added. On top of that, we are going to heat. On top of that, we add an acid, which is usually sulfuric acid, and look at the background blue aa raha hai na? So most of the cases we add methylene blue. That is what you need to know. So class has asked mnemonic was our protocol here. So now tell me guys, that whenever you see this, can you see what is the color that the organism has got? Kya color aaya? The organism has got a beautiful red color. Why? Because the red color is because of carbonyl fustian. And what is the background? The background has come out to be blue. So there are two colors. If they ask you, what is the organism? The organism has come out to be red and the background has come out to be blue. And this is a question that once came in the exam, in which they did not give you the photo. They just gave you a fill in the blank question. Organism is dash, background is dash. So now only that person can answer who's seen the photo earlier. Organism is red, background is blue. Okay, I'll come to all these other features, but tell me one thing, how much of TB should a person be having that the person is able to show this positivity? And that is 10 to the power 4 acid-fast bacilli per ml. 10 to the power 4, okay. And you will say that if ma'am you are, who sees this? Either it's a pathologist or a microbiologist. Someone with a microscope can see this, hai na? So you will say that ma'am, how much time do you spend on every slide? Agar to you saw even if one TB was seen, I will tell the patient you have TB. But I'm not seeing any bacteria. Maybe it is not TB. How many minutes should I keep seeing the slides? So remember, they say that usually you should view every slide for 10 minutes. 10 minutes mein on an average, how many fields can a pathologist see? A pathologist can see 100 fields. And guys, what is the sensitivity? 10 to the power 3 to 10 to the power 4. 3 matlab 1000, 4 matlab 10,000. So basically I have made a rule of 10, 100, 1000, 10,000. And what is that? You have to see every slide for 10 minutes. This means you have to see 100 fields, and the sensitivity is going to be 10 to the power 3 to 4, or you can do double also. If you don't want to learn 10 to the power 3, you can just learn 10 ka square kara, so you get 100, and then you add it almost in another league, that is 10,000. 10, 100 and 10,000 have to be the acid fast bacilli per ml. So now I want to ask you that why is this acid fast? What is the reason for acid fastness? I had already told you the reason is mycolic acid. The reason is mycolic acid. So tell me one thing, that there is something called MTB, but uska ek cousin brother hai, that is something called M. bovis. Okay, so this is what I'm going to come to. Remember MTB, when you will look at it, it's going to look very beaded. It's just not going to be, you know, like red color rod like this. No, it will look a little beaded like this. That is going to be MTB, whereas M. bovis is going to be totally smooth. That is the appearance that you have. I will talk more about MTB and M. bovis in a while, but I want to ask you guys that if you see this, are you like very sure that ये TB ही है? Can it not be anything else? Can it not be atypical TB or non-tubercle mycobacteria? हो सकता है मैम, maybe it's one of those rare mycobacteria. So now what will you do? So now I want to come back to the protocol and ask you, when you were doing class has asked mnemonic, how much of acid were you using? TB के लिए कितना acid? So we had done TB के लिए T20. 20% H2SO4 is what we were using. So please note that MTB is acid fast, means with how much of acid it will not give away the color with 20% also, T20%. Not only is it acid fast, MTB is also alcohol fast. If you would have used 95% alcohol, then also it would have retained its color. When I say it is acid fast, fast matlab chalu, it is not going to give away its color. Once it gets the red color, it will say, I will keep my red color. You put acid on top of me, you put any alcohol on top of me, mera red color to rahega. That is what is acid fast. So whether you call it acid fast or you call it alcohol fast, it's both. And that is what I want to tell you, that if you're talking about TB, versus if you're talking about atypical TB, the ones that I will teach you in the end, TB is going to be both. TB is fast, it is acid fast also, and it is going to be alcohol fast also. When you're talking about atypical TB, everyone is acid fast, but it is not alcohol fast. This is a very important difference that you have to know out here, right? So now once you have visualized this, you will say, ma'am, is there any grading? Can I know that this person has thoda sa TB or too much TB? Basically, lower load of the bacteria or high load of the bacteria. Can you do any grading? Yes, and this is a tough grading, but I will simplify it for you. Dekho, if you do not see even one organism totally negative, khatam, patient doesn't have TB. Even if you see a single organism, even one, that means the patient has TB. No doubt about it. Even if there is one TB seen, one red color organism seen, it is TB. Okay. Now you will have to do, how many fields did I tell you have to visualize? Minimum kitna karna hota hai? 100 fields na? So 100, 100. So pehle you will do 100 fields. In the first one I am getting 1 to 9 bacilli. I will say theek hai, ye 1 plus hai. This is scanty. Positive matlab scanty. If I say 10 to 99, that becomes 1 plus. 1 plus matlab thoda sa zyada hai. This is what I'm seeing. See, 1 to 9 bacteria in 100 fields. 10 to 99 bacteria in 100 fields. 99 matlab 100. Now when I say 100 in 100 fields, this means it's 1 in every field that I'm seeing. So now the next two categories are per field, per field. Now in every field I am seeing 1 to 10, and the next in every field I am seeing more than 10. That becomes 2 plus and 3 plus. So now let us all of us write this once again. Simple, what I'll do, negative ko to negative karo, side karo. First I will see 100 fields, per field, per field. 100, 100, per field, per field. You can learn it as 1 to 10, 10 to 99, again 1 to 10, more than 10. Simple. 1 to 10, more than 10, 1 to 10, more than 10. 100, 100, 1, 1. Abhi bhi clear nahi hua? Dobara karo. 100 fields, 1 field, 1 field. How I have learnt it? 1 to 10, more than 10, 1 to 10, more than 10. That makes it what? That makes it scanty, 1 plus, 2 plus, 3 plus. I know it's tough. एक दो बार just write it on your own, हो जाएगा. That is the grading. But what am I using? Till now, I have only been focusing on the ZN staining. Now I'm going to ask you a question again from General Micro. Was there a fluorescent stain that I used for TB? One was auramine and the other was rhodamine. I want all of you to note over here, rhodamine is for Mycobacterium TB. Because way back in pathology, I have taught you something called rhodamine and that was for copper. So remember, rhodamine is going to be for Mycobacterium, rhodamine is for copper. So chalo, this is very, although the problem is ke dekho, fluorescent microscopy, so I can see it's shining, it's fluorescent, but it's very expensive. Still, we say that as per our programs, this is the recommended screening method. Why? Because it's sensitive and rapid. Sensitive and rapid hai, otherwise it's very expensive. So recommended screening method asked to you for your MCQs is going to be fluorescent. Auramine, rhodamine, MM wala for Mycobacterium TB. We are ready for the culture. I know you guys are very scared of culture media. I've got you a lot. Of course, in detail, you just have to read once. So, baki sare we will do names in the end. Main kar lete hain. The egg-based culture media are the Lowenstein-Jensen medium and the Dorset egg medium. In dono mein egg hota hai, that is why they've called egg media. So my focus is Lowenstein-Jensen medium. Let me show you everything about it. What is it? Lowenstein-Jensen medium. And how will I learn the composition? For learning the composition of LJ media, you have to look at a beautiful English breakfast. What all will you have in an English breakfast if you go abroad? You will have nice eggs. So nice boiled eggs. So this has number 1 coagulated hen's egg. Along with that, you will say, can you sprinkle a little bit of salt on the eggs? So this has mineral salt solution. Then you will say that I want some asparagus and some green vegetables. Yes, I'm giving you some green vegetables. Asparagine, not asparagus. I'm giving you asparagine over here and I'm giving you malachite green. So that is your ideal breakfast, right? English breakfast. It is going to have eggs. It is going to have salt on top of it. It is going to have asparagus and asparagine, because asparagus is a plant na, so green color plant, asparagine and malachite green. That is why you will see LJ medium ka background green hai, because you have added malachite green. And what colonies are you getting? Do you want to zoom into these colonies and see? Look at these. They are rough, buff, tough. It's like a rhyme. This is going to be a rough colony. It's going to be rough, buff, tough. Buff means, buff refers to cream color. See, it's not coming across as white, pearly white, nahi hai, cream color hai. And tough because you can't remove it. So what did I say? This is going to be a rough, buff, tough colony as you see over here. That is what you see on the LJ media. But the problem is that this is taking a lot of time. This growth takes 6 to 8 weeks to grow. Although the sensitivity is good, 10 to 100 bacilli per ml sputum. If I ask you for the light microscopy, it was 10 to the power 4 acid fast bacilli, 10,000 yaad hai, per ml sputum. Whereas for LJ media, it has come down to 10 to the power 1 to 10 to the power 2. So basically 100 acid fast bacilli per ml. Till now this has happened. So what have I taught you? I have taught you this is the egg based media, LJ, and then there's another one called Dorset egg. Others you just have to go through names one. For blood based, there is something called the Tharshi media. There is serum based, which is known as the Loeffler's media. There's potato based, P for P. This mnemonic I can tell you. Potato based is going to be Pavlovsky medium. And finally, you have the liquid media. If you ask me out of these, what are the things they're going to ask me, ma'am? Egg based to पूछेंगे, because that is what they definitely use. LJ media to use hota hai. Potato ka Pavlovsky they can ask. That is again something that they will put up to all of you. Potato Pavlovsky, and liquid media they will be asking. What are the liquid media? Okay, think of some liquids. What are the two most liked liquids in the country? Either it is coffee, right, or it is alcohol. These are two very liked liquids. तो एक coffee का नाम रखते हैं, एक alcohol का नाम रखते हैं, ठीक है? So please remember, there's a very famous coffee known as Brooks coffee. तो Brooks is Middlebrook. Middlebrook जो medium है, Brook will remind you of coffee. Coffee is liquid. Then people who are fond of wine, India की सबसे favorite production of wine, Sula wine from Nasik. So that is going to be liquid again. And if you can learn Proscure and Beck's, the liquid culture media. So one coffee, one wine. Middlebrooks and Sula, both of them are liquid. So these are liquid culture media. And what do you get? You get long serpentine cords on liquid culture media. You get long serpentine cords. Why? Because cords क्यों आएगा? Because of something called as cord factor. That is how I have learned it. Cord is because of cord. Didn't I tell you in the first slide TB has cord है ना? तो cord is because of cord. All these culture media are done, but this is taking a lot of time. 6 to 8 weeks. Who will wait? The patient will have so many problems if you wait for a report for 6 to 8 weeks. So you definitely need some newer methods, some automated methods which can give me a report very quickly. 8 to 14 days, not very quickly also, but better than 6 weeks. 8 to 14 days. That is Bactec MGIT. Firstly, Bactec will tell me kuch bacteria related. MGIT will tell me, it is I'll tell you the full form. It is M for, of course, has to be mycobacterial growth indicator tube. Mycobacterial growth indicator tube. So if you split the word, you will say, okay ma'am, over here you must be using a tube. In that tube, your mycobacterium is going to grow, and as soon as that mycobacterium will grow, the machine will give you an indication कि हाँ, mycobacterium has grown. How will the machine give you an indication? It will give you an indication by some shiny, some color, some fluorescence. So this is a fluorescence method. So what does this use? It uses one of those Middlebrook 7H9 मतलब Middlebrook. Brook must be that liquid wala media. Brook's coffee na. So Middlebrook must be that liquid wala media. So what happens over here? For example, if I draw this as one of your test tubes and I say right now, this has your Middlebrook, I will write 7H9, okay? This has your Middlebrook 7H9. To this you have added your TB, whatever sample you sputum या जो भी sample आया था, and right now does it have oxygen? है ना? Right now it has oxygen, and because it was a fluorometric technique, of course there is some fluorescent dye also present. You know, no one's going to ask you what all it has. I'm just telling you one simple concept. So now the concept says that till the time oxygen is present, the fluorescence will never show. Fluorescence will shy away. Fluorescence will be quenched. However, when Mycobacterium TB will grow, you know, Mycobacterium TB is aerobe. Mycobacterium TB will utilize all the oxygen. It will utilize all the oxygen. The oxygen is used up. Now, when the oxygen will be gone, then this fluorescence will say, oh wow, party time, oxygen is gone, let me show up. This means there is gonna be fluorescence that will be seen. There is a fluorescent indicator that is noted. That is why you called it an indicator tube. So simple, in the beginning, oxygen is always there. Oxygen tha. So since oxygen was there, fluorescence said, I will not show. But now Mycobacterium TB has used all that oxygen. Oxygen dheere dheere chala gaya. Now the fluorescence will start showing. So simple. If Mycobacterium TB has grown, you will start getting an indication. Simple. That is why you called it Mycobacterium growth indicator tube. If Mycobacterium has grown, you will start getting an indicator. That is the Bactec MGIT. Similarly, you have other machines, guys. There is something known as Bacti-Alert. Bacti-Alert works on color change. It is a colorimetry based method. There's something called Versa-Trek. देखो, trek करने के लिए you need a lot of strength, है ना? तो Versa-Trek is gonna measure your pressure changes. It measures the pressure changes, right? So remember, Bactec Alert is color based and Versa-Trek is going to be pressure related changes. Also, if you ask me, are they only giving you the diagnostic ability? No. If you want to find out that is my tubercle bacillus resistant to pyroxenamide, to rifampicin, I want to find out the sensitivity and the resistance of the drugs, I can do that also. Let me show you a machine. This is what a Bactec machine looks like. This is how the tubes, the MGIT tubes look like. So not only can they tell you about the diagnosis, but they can also give you information about the resistance. What else? Apart from this, you have some typing methods also. Typing methods में कुछ genetics आएगा, ठीक है? What are the genetics that you have? One is RFLP. What is RFLP? RFLP is a type of PCR that is restriction fragment length polymorphisms. And if you have read your way back chapter of your pathology, you have seen in PCR, I have told you, restriction fragment length polymorphism is something which is seen, and they will ask you, okay, whatever is going to be the PCR ka methodology, just tell me what is your target sequence of strain. That is the question. That is IS6110. IS6110 is the target sequence of strain that we have, but सुनने में तो it's looking nice, but there's a problem. It cannot differentiate active TB versus latent TB, and that is very important because in a country like us, more than half the people have latent TB thanks to the exposure, right? So remember that we cannot differentiate between active and latent TB. That is something which is very important for RFLP IS6110. Okay, now there is something which I'm going to tell you, something called as line probe assay. ये भी दोनों काम करता है. Diagnosis also it will tell you, resistance also it will tell you. But now what I'm going to tell you, that is Gene Expert, and that's the correct spelling, XPERT. Gene Expert. This is something which we are using. So I won't teach you here. I will teach you directly with the help of this picture. This is the picture of a Gene Expert. If you're smart enough in the exam, you will be able to read also. On the machine, it is written Gene Expert. What is Gene Expert? Gene Expert is a type of CB-NAT. मतलब कुछ तो जेनेटिक्स है मैम. आई कैन सी देर इज अ कंटेनर में सैंपल होगा, पेशेंट का सैंपल, सम स्प्यूटम और समथिंग मस्ट बी देयर, एंड आई कैन सी इट हैज अ नाइस बारकोड एंड अ कार्ट्रिज टू इट. एग्जैक्टली, CB stands for this is a cartridge based, and NAT we already know it is a cartridge based nucleic acid amplification test. It is a cartridge based nucleic acid amplification testing. So it is a genetic testing. What is this useful for? It is useful not only for the diagnosis, it is also useful for finding out the rifampicin resistance. It is also useful for finding out the rifampicin resistance. If I ask you a little bit of pharmacology, rifampicin resistance is because of which gene? It is the RPUB gene in the bacteria that causes it, है ना? So if the bacteria is having a problem in RPUB gene, then the bacteria will be resistant to rifampicin, right? So remember, Gene Expert is for knowing diagnosis as well as the resistance. So why am I talking so much about it? Because इसका जो turnaround time है, TAT, guys, TAT, any test, like you must have seen when you go and give your blood sample or any culture sample or anything to a lab, on the reception they tell you we will be able to give you the report after 5 hours. Our turnaround time, TAT means turnaround time. This means you've given us our sample, we will complete it within 5 hours, because our lab protocol is turnaround time is 5 hours. Can you imagine, iska turnaround time is only 2 hours. And if you ask me in terms of its sensitivity, it is 88%, but its specificity is 99%. So it's a damn good test, as I can see. And if you ask me in terms of what is the detection limit, how many bacteria should be there? So as per the latest guidelines, they say 131 organisms present per ml. If this is the limit, then you can detect it via Gene Expert. So every word is important for the diagnosis, for the resistance. It has a good sensitivity, specificity. Detection limit is 131 organisms per ml. Everyone's okay with the genetics as well? Yes, the typing methods, RFLP, but cannot differentiate active from latent, Gene Expert, and line probe assay. Okay, next we are going to go on to what is the diagnosis of latent TB. Latent TB means, which most, as I said, many of us might be having. And latent TB is of two types diagnosis tests that you can do. There is something called a Monto's test and there is something called IGRA. These are the two tests. Monto's thoda cheaper hai, IGRA is a little expensive. So first, Monto's. I think everyone knows Monto's test or tuberculin test is this very, very famous test which is using an antigen. What is that antigen? That antigen is PPD, made out of TB only. It is a purified protein derivative. It is a purified protein derivative. What is the procedure? You're gonna take 0.1 ml of PPD and you are gonna inject it intradermally in the flexor aspect of the forearm. As you can see, you took only 0.1 ml of that PPD and you have injected it intradermally in the flexor aspect of the forearm. Now you're going to take a reading after 48 to 72 hours. And now what is the reading that you'll take? So injection to de diya intradermal, 0.1 ml of PPD injection you've given. Now when you look at the reading, so see, you have given an injection over here. Patient came to you after 48 to 72 hours with this. So how have you identified? So see, this is the forearm. How have you kept the scale? You have kept the scale perpendicular. So please remember that you have to do the measurement which is going to be perpendicular to the long axis of the forearm. Don't start measuring it like this. No, perpendicular to the long axis of the forearm is very, very important, right? So what do you have to measure in this and what do you have to ignore? So please note that if you zoom into this, you will see that there is one area which looks thickened or nodular and another area just looks a little red around it. So please note that what you have to measure is going to be that thickened area, that is induration, and what you have to ignore is that redness, that is erythema. You don't have to measure the redness because redness can occur occur because of any other injection also, local site injection, right? So it's not going to be the redness that you measure. What is the final interpretation? If you get an induration of equal to more than 10 mm, this test is positive. Less than 5 mm is negative, and anything in between is going to be equivocal. So like I said, less than 5 mm is negative, between 6 to 9 is a question mark is what you can say, equivocal. Equal to more than 10 is positive. Although nowadays many of them are saying that in our country more than 5 is only positive, but it's yet to be documented in textbooks. Now on that note, you would want to know that how is this, what is that positivity telling me? Positivity is telling me some kind of an infection. It could be a present infection also, it could be a past infection also. That is why this is just a screening test, right? So present or past infection. When do you get a false positive? If recently person had received a BCG vaccination, that is when a person can have a false positive reaction, or if there is an atypical mycobacteria or even non-tubercle mycobacteria can also result in a false positive. When do you get a false negative? If it's a very early case of TB, maybe see what is Montu's test? Montu's test is basically your immunity. Which immunity? Go back to your pathology and your immunology lectures of micro and path. Tell me, you know that Montu's test is a type 4 hypersensitivity reaction, which means it is a cell-mediated hypersensitivity. Guys, it is the cell-mediated. It's your immunity which has to work. So what if you have an early case of TB? Right now, your immunity has not started working. Or what if there is a case of low immunity? There is a low immunity in the body. For example, if it is a case of HIV. If there is low immunity in the body, this means the Montus shall not come positive at all. But that's the wrong case, right? This is a false negative situation that has occurred. So please note, this is the interpretation that you have. But this was just one test that I told you. I told you one test is going to be Montus and the other is going to be IGRA. So what is IGRA? First, you'll have to know the full form. I'm sure all of you have learned that TB shows you granuloma. Let me ask you your basics. TB shows you granuloma. Immunology yaad hai? Granuloma is because of which interferon? So we have studied gamma for gamma. Granuloma is because of interferon gamma. And interferon gamma, if I can measure, that is known as interferon gamma release assay. So in this test, I am going to measure the release of interferon gamma by which machine? By an ELISA. So this is the protocol of this particular test. You are going to measure the release of interferon gamma by ELISA. Now tell me, go back to your immunology. Which cells release interferon gamma? It is T helper 1 cells. This means what is the sample that you're going to take from the patient? You will have to take the T lymphocytes of the patient. Dekho, sab kuch, if you know your basics, you know. In IGRA testing, I will take a simple blood sample from the patient. In that blood sample, I am basically going to go ahead and take the T lymphocytes. If I have the T lymphocytes from the patient, I will have to give some antigen to stimulate the T cells. And I am going to expose it to antigens, which are 3 antigens, 6, 7, 10. What is 6, 7, 10? It is ESAT6, TB 7.7 and CFP 10. So remember, ESAT 6, 7.7, CFP 10. There are many other antigens also. These much also you can learn is more than enough. So 6, 7, 10, and that will stimulate your T lymphocytes. If you have been exposed to TB, that will release interferon gamma. You will measure it by ELISA. This has an advantage. See, till now the problem was that you were not able to differentiate. Finally, you have a technique where you are able to differentiate. So now this has one advantage and one disadvantage. The advantage is, what is the advantage over Montus? The problem with Montus was that it was giving you a false positive in BCG vaccination. This is not going to be positive in BCG vaccination. So the advantage is that it is going to come out negative in BCG vaccinated people. So that's an advantage. But what still remains as a disadvantage is that this is a test for latent TB. It cannot differentiate latent from active. So that you need a gene expert or a nucleic acid method testing for that. So these were the methods that you have for the two, you can say the two latent TB wala things. Now there is a test which is hardly done nowadays, but for our MCQs I'm telling you, there is an MPT64 test. What is MPT64? It is a transdermal patch test. Transdermal patch test. So what they do, they take a patch and on that patch they have the MPT64 antigen. And with that MPT64 antigen, that is basically placed transdermally in your body. And once that is placed transdermally, you will be able to see the reaction, right? You will be able to see the induration and the erythema. So basically, Montu's test ka transdermal version. Okay, you will be able to see the induration and the erythema. So this is positive only in active TB. Montu's was even positive in latent TB. So this has a 100% specificity and a 98% sensitivity. But since it is transdermal, it can be a little tedious to perform. So you just have to know it is an MPT64 assay, which is a transdermal method for active TB. Then in the beginning, I told you there is going to be something called LAM assay. What was LAM? LAM was lipoarabinomannan. And this was a testing I told you will be important in the urine, and especially in patients who are HIV positive and have TB. Also, please remember, if someone asks you what is the principle of the lipoarabinomannan assay, that is going to be immunochromatographic testing. Now, since we've discussed so much, why don't we discuss the vaccine also? We all know the famous vaccine, that is BCG, Bacillus calmette-guerin. And as per the WHO, you know what strain is used. Danish 1331 strain is used over here, isn't it? Where is the central BCG lab in our country? I hope you all know it's in Chennai, yes? And what is the dosage that is administered? What is the site? It's in front of you. The dosage we all know is a 0.1 ml, and the site is going to be here above the insertion of the left deltoid. So left is basically considered for our standardization, so that the scar is available in everyone on the left and it becomes easy to screen the population. Otherwise, there's no left-right mandatory thing, only for standardization. So remember, 0.1 ml of this Danish 1331 strain is injected above the insertion of deltoid, and that is BCG. However, you must know that although we say that once we are injected with BCG, we have immunity for lifelong, but a lot of studies say that the immunity is 15 to 20 years approximately. After that, you should know the scar is going to come up somewhere in approximately 6 to 12 weeks, depending on the immune response of the person. And because I told you that after BCG, MON2s can be positive, so this is a question which many people ask me, that someone got BCG vaccinated recently, so MON2s can come positive till how much, what is the duration? Even after 8 to 14 weeks, the MON2s can be positive. After 8 to 14 weeks, it can come out positive. So India mein to a lot of people are definitely going to be positive MON2s, because it's definitely everyone gets a BCG vaccination. Okay, so now you know BCG, this wala use to I knew, everyone knows this BCG vaccine. But what else is the recent BCG vaccine used? Guys, there is a strain called the oncotype strain, which is used for superficial bladder cancer. They are now trying to utilize it for the superficial bladder cancer and the early stages of bladder cancer, because we all know urinary bladder. Urinary bladder cancers have a very bad prognosis. They are very invasive. They quickly spread and patient does not have a good survival. So only in early cases we can try this, and this will come as an intravesical injection of BCG. So the oncotype strain is used for that. Mycobacterium TB ho gaya. Can we move on to MOT? What is MOT? Mycobacteria other than tubercle bacillus. Mycobacteria other than TB. So mycobacteria other than TB makes it MOT, or you want to call it non-tubercle mycobacteria, atypical mycobacteria, your wish. Environmental mycobacteria, because environmental matlab they are reservoirs happening in soil and water. This is not going to be a TB ki tarah human to human thing. This is a reservoir soil to soil thing. Now tell me a point which I've already told you. Acid fast hai? Of course, everything is acid fast, but is it alcohol fast? No. That is what I told you, the difference between TB and MOT. That acid fast is going to be everything. Both of them are going to be acid fast. But when we are talking about alcohol fast, it is only TB which is alcohol fast, and MOT is not alcohol fast. What is the classification that we have? Classification is a famous one known as the Runyon's classification. Runyon's classification says photochromogens, cotochromogens, non-photochromogens, and rapid growers. What is their definition? Photo means some pigment. This and pigment to production hoga. There is definitely a pigment production that we are gonna have. But please note that photo means light mein hoga. That pigment production is only gonna occur in light. Scoto obviously means dark. So there the pigment production is, now this is where the students don't read carefully. Pigment production still occurs in the light and the dark. So some people feel only in the dark. No. Photochromogens means only in the light, but scotochromogens means is dark and the light. Non-photochromogens basically means that there is no pigment production that is going to occur. And what is rapid growers? Do you remember I told you TB ke liye ek bacteria culture hota hai, that is LJ media. TB was taking how much time to grow? 6 to 8 weeks to grow. This also takes a lot of time to grow, but rapid growers will grow within 1 week. 1 week means 7 days. they are going to grow very, very rapidly. So what did I say? All of them are going to grow on LJ media, but these will take a lot of time to grow. Rapid growers will grow within 7 days, and on LJ media, guys, they produce a very famous orange color pigment. LJ media pe, they are very, mott is known to produce an orange color pigment, and all of them are also catalase positive. This I've already told you. Every mycobacteria, TB or mott, everything is catalase positive. Coming back to the Runyon's classification, this is a classification which most student leaves. I'm going to simplify this for you today. Photochromogens. Whenever you get a photo clicked, nowadays you have so many filters, na? So you have so many filters means you're going to put a mask on your face. Next time you get a photo clicked, you put a mask, and you can see how this girl has transformed with filters on her face. They start looking like different people altogether, hai na? So mask ya filter laga liya. So I have named this girl Simran. Why have I named her Simran? I'll tell you. Simran is putting a mask for a photo. Means M for Marinum, A for Asiaticum, S for Simye, Simran, because there are so many S's. So Simran is the girl who has put the mask, so that is Simye, and K for Kansasi. And you can see in all of them there are respiratory infections that are occurring and scar formation. Some में lung involvement hai, but that Marinum is sounding only like marine water. That is going to cause swimming pool or fish tank granuloma. So marine is what you will think of. Swimming pool or fish tank granuloma, and that is a skin problem with a warty lesion. The skin is going to show you a warty lesion. So please note, marine will remind you of the swimming pool or the fish tank granuloma. So Marinum, Asiaticum, Simye, Kansasi, all of these are photochromogens. What is the next one? Scotochromogens. Scoto ka kya matlab tha? Dark. And how I have learnt is, God, I'm very scared of dark. I personally telling you, God save us from the dark. May God save us from the dark. So I am very, very scared of dark. So dark matlab scotochromogens. God means Gordone. God save us. Save means scrofulatium and sulgi. Okay? So God for Gordone, save us, scrofulatium and sulgi from the dark. They are going to be scotochromogens. Please note, God has provided you water for free. Air and water, everything is free. So God has given you this scotochromogen for free. Mycobacterium Gordone is a tap water scotochromogen. So many a times you might find this in gastric lavage samples because this is a tap water scotochromogens. Okay, next you have scrofulatium, which is of course going to cause scrofula. This sulgi sometimes is a little idhar-udhar. Means sometimes it can be scotochromogen, which is at 37 degrees Celsius. Sometimes it can be photochromogen also, that is at a 28 degree. Not very important information, miss karna hai kar do. Temperatures are not at all important. You have to know it can go here and there. So remember, God save us from the dark. Now let's come to the next one. So the next one, I've not got you any mnemonic because now everything that is left, the non-photochromogens will have Mycobacterium avium. There's going to be Mycobacterium intracellularly. There is ulcerins and xenopi, whatever is left. So simple. In photochromogens, you had mask, Marinum, Asiaticum, Simye, Kansasi. In scotochromogens, you have God save us from the dark. So Gordone, scrofulatium and sulgi. Whatever is left is over here. Mycobacterium avium, out of this entire mott family, is the most important for causing granuloma. So TB was causing granuloma, and this Mycobacterium avium also causes granuloma. In fact, Mycobacterium avium and intracellularly together are known as Mycobacterium avium complex, for which I want to tell you something. Mycobacterium avium complex. When I say this word, I say it as Mycobacterium, then avium complex, but everyone learns it as MAC. Now tell me something. MAC, girls will love this mnemonic. MAC is a very famous and very expensive makeup brand that we have. Hai na? So as soon as I say MAC, you are reminded of lady, because this is a question. There was a syndrome known as Lady Windermere syndrome. So if you Google it out, it used to be a Victorian play. And in that Victorian play, they had this character called Lady Windermere. Lady Windermere was basically a female who was very, very royal and very sophisticated. And I don't know which form of sophistication it was, but she never used to cough. Whenever she used to feel that she has to cough, out of that sophistication and mannerisms, she never used to cough. So what she used to do was, she used to suppress her cough. And if she was suppressing her cough, that meant that the secretions were never cleared. The secretions were always accumulating inside. They were always accumulating inside the lungs, and this was resulting in bronchiectasis. There was a problem in the lung happening, and then there was a risk of infection. That was MAC. Only I'm telling you this play story for your interest. Otherwise, you should know MAC matlab makeup. Makeup ladies में करेंगे तो lady Windermere syndrome is associated with MAC infection. This is done. Now, who causes Burulli's ulcer? Of course, no doubt about it. Burulli ulcer will be caused by ulcerins only. And let me tell you, this is the only one which produces an exotoxin, the only mycobacteria to produce an exotoxin, that is mycolactone. That is mycolactone. So please note, ulcerins has of course Burulli's ulcer, and it is the only one. Ulcer क्यों करा रहा है? Because of mycolactone. Starting last one, xenopy. Xenopy anyway means like xeno, we always refer to something, animals outside of this world. That means that this is the one that can grow at very, very high temperatures, and this is the one that is going to be found in hospital waters. That is xenopy. So the non-photochromogens are also done. एक और आता है. There is something known as, you can call it MIP, it's my favorite question, Mycobacterium indicus prani. Because it sounds funny basically. Mycobacterium indicus prani. If you see, it has a lot of eyes. Mycobacterium indicus prani. So it is used as an immunomodulator agent. It is an immunomodulator agent. You know what they do? With this, they make a vaccine called the MW vaccine. And nowadays that vaccine is being coming out to be useful for leprosy. So Mycobacterium indicus prani is an immunomodulator, MW vaccine for leprosy. Finally, all of them are done. I'm left with the last variety, that is rapid growers. Rapid growers means the ones who grow very quickly within 7 days. So tell me, who are the people who become overnight famous? I'm not talking about all the social media influencers. I'm talking about, what do you think? People who become famous overnight, viral हो गए. Number one, either they are very, very fortunate people, very lucky people, है ना, तो Mycobacterium fortuitum, or they are very, very smart and hardworking people, smart for smegmatis, or they are just simply very chalu, clever kind of people. So chalu, clever means chelone, or they are so lucky in life that they were just sitting free like the influencers and doing and still getting famous. So that is flea. So please remember, Mycobacterium, who are the people who rapidly become viral? Fortunate, smart, chalu and free people. Fortuitum, smegmatis, chelone and flea. These are going to be the rapid growers, and especially chelone. This has a risk of post-surgery abscesses. Like there'll be a person who's undergone a knee replacement or a hip replacement, usme abscess ho gaya, and that is growing very, very fast. So the hint in the question will be this, which is a rapid grower. On a LJ media, it ended up growing in 6 days, 5 days, less than 1 week. You know you're talking about a rapid grower. So on that note, you are done with the entire MOT family also, and I hope you guys have been able to analyze the mnemonics. Photochromogens ke liye what do you have? You have mask, Marinum asiaticum, Simiae and Kansasi. For scotochromogens, God save us from the dark, Gordone, Scrophulatium and Sulgai. All the others are non-photochromogens. And what is rapid growers? So fortunate people, fortuitum, smart people, smegmatis, chalu people, chelone, and free people, flea. These are the rapid growers. Done with this, the last mycobacteria that we are left with is the Hansen's bacillus, and Hansen's is known as Mycobacterium leprosy. Now, the first two lines, it is obligate intracellular, obligate aerobe, means it will always need oxygen and it will always be inside the cells. Anything that grows inside the cells means it cannot be grown on culture, it cannot be grown on culture media, it can only be grown in animals, and that is what I'll tell you. This is actually grown on animals, not in culture media, because you need cells for their growth. How does it spread? Most common nasal droplet followed by contact and even milk. So we all know that there is a very famous leprosy classification that we use. There are lots of Indian classification, Madrid classification, which you study in skin. Dermatology में बहुत पढ़ोगे, but the universal one is the Ridley-Joplin classification. There is an LL pole and there is a TT pole. LL refers to lepromatous leprosy. LL refers to lepromatous leprosy. TT refers to tuberculoid leprosy. TT refers to tuberculoid leprosy. In the center, you just have borderline. In the center, you have borderline leprosy. And in between the two, now you can say borderline lepromatous, borderline tuberculoid. So simple. So one end is only lepromatous, one end is only tuberculoid, one end is borderline in between. So in between these two, there is borderline lepromatous. In between these two, there is borderline tuberculoid, and that is going to be the Ridley-Joplin classification. So my first concern is to be able to make you differentiate between these two poles. That is what is important, and the difference is on the basis of immunity. Please note that your LL has a lot of bacteria. It is multibacillary. Why? How much bacteria? There's an index. There's a bacteriological index. We say very, very high load, 4, 5, 6 plus. Too much of load. So now tell me, when there are too many bacteria in my body, means kya problem hua hai? Has my immunity been able to fight? No. There are so many bacteria in my body that this means that my cell-mediated immunity is somewhere low. That is why so many bacteria are there. And I know which is the skin test that takes care of. Jaise TB mein there was Montu's test for cell-mediated immunity. Leprosy mein there is Lepromin test for cell-mediated immunity. But when I'm saying that the cell-mediated immunity is low, this means the Lepromin test is not gonna come out. It's gonna come negative. So what have I learned up till now? This is gonna be lepromatous leprosy. Is gonna have a lot of bacterial load, is gonna have a low cell-mediated immunity, is gonna show you late nerve lesions. The nerve lesions are gonna come out very, very late, and the skin lesions are gonna show you leonine facies. They are going to show you leonine facies with the alopecia. This is something that I will be showing you. Have a look at this picture. Can you see these nodules? And these nodules have basically transformed it into lion-like facies, leonine facies. And if you look at the eyebrows, you can definitely appreciate there is eyebrow alopecia that you can see in these patients. So yes, coming back, you have everything LL. This will help you in derma also. Load is very high because the cell-mediated immunity is very low. Wo low hai, matlab Lepromin is not gonna come. The nerve lesions are very, very late. Clinically, patient has leonine facies with alopecia. So tuberculoid is gonna become opposite. Tuberculoid is now gonna be what? It is gonna have less of bacteria. Paucibacillary means hardly any bacteria is there. Why? Because the cell-mediated immunity is perfectly fine. If it is perfectly fine, this means the thing is going to be positive. Lepromin test is positive. Nerve lesions kya hoga? Ab udhar late tha to yahan early hoga. Over there were lots of lesions. Over here there are very few lesions that you're going to see. So everything becomes opposite. The table is not over. Please note, when you're talking about the CD4, CD8 and humoral immunity wala concept, tell me one thing. In lepromatous leprosy versus tuberculoid leprosy, CD4 upon CD8, pehle to you will have to tell me the normal ratio. The normal ratio is 2:1. Everyone remembers? Okay. I can see tuberculoid leprosy weight is 2:1 because the cell-mediated immunity was normal. CD4 में कोई problem नहीं था. In lepromatous leprosy, the cell-mediated immunity was low. If it is low, I can see the ratio has become ulta. It has become 1:2 because the CMI was low. Now tell me, if one immunity is low, this means the other immunity will wake up. If cell-mediated immunity is low, this means the humoral immunity, antibodies ज़्यादा बनेंगी. That is, body will compensate. Na? Humoral immunity will be more, whereas in case of tuberculoid leprosy, the humoral immunity is also normal. CMI also normal, humoral also normal. Now tell me, tuberculoid sounds like TB. So will you see TB ki tarah lots of macrophages and lots of giant cells in TB wala tuberculoid leprosy? Not in lepromatous leprosy. And on the same path, if you ask me, how are you going to do the diagnosis of this? What is the diagnostic modality? I will take the specimen. How many specimen? We have to take 6 smear samples, out of which 4, 1, 1. 4 have to be taken from the skin. Skin of where? You can see forehead, then you can say cheek, then there is going to be chin. So 1, 2, 3, there is going to be forehead, cheek, chin. There is also one from here because I'm talking about the face now. There is 1 teen ho gaya. The 4th one is going to be here, that is the earlobe, and the 5th one is going to be from the mucosa. So there's going to be forehead. Sirf head and neck I'm talking about. The forehead, the cheek, if you want you can include the earlobe, and the chin. 4 ho gaye. The 5th one is from the nasal mucosa, 5, and the 6th and the last one is going to be from the buttock. Repeating: forehead, cheek, earlobe, chin, nasal mucosa, and then in that order, after that you have the buttocks. Okay, so this is going to be, you're going to pinch the skin and you're going to make a small incision. Slit skin smear is taken. Also, I told you some nerve lesions can be there. Agar nerve lesion hai, to you take a biopsy from the nerve also. What are you going to see? I just told you, if you're dealing with lepromatous versus tuberculoid, where will you see macrophages and where will you see giant cells, granuloma wale features tuberculoid mein? What will you see in lepromatous? You will see the presence of Virchow cells. Remember, lepromatous leprosy shows you the presence of Virchow cells. What are Virchow cells? They are big macrophages. Virchow cells are nothing but macrophages, and these macrophages, so this is a macrophage that has engulfed the Mycobacterium leprae. So how will it look like? Look at this. Lots of, what did I tell you? Lepromatous leprosy mein, ma'am, the load is going to be very, very high. Lots of bacteria are there. So look at these bacteria. They are in bundles. So these have identified lots and lots of bundles. You know what these bundles are known as? These bundles of the bacteria are known as cigar bundles. Like cigar or BD comes in bundles. So it's known as cigar bundles, or it is also known as globi. It is known as globi or cigar bundle. So I can say this is a very big macrophage, and that macrophage has basically engulfed all of these cigar bundles. So this entire cell out here is known as a Virchow cell. This is what a Virchow cell is. So remember, Rudolf Virchow was the scientist. So it's a macrophage which has eaten up all of those cigar bundles. That is all lepra bacillus. And this is seen in lepromatous leprosy. It is seen in lepromatous leprosy. But what stain? You will say, ma'am, for full Mycobacteria, you have Zn stain. But when I'm talking about Mycobacterium lepra, 1, 2, 3, 4, 5, what H2SO4 percentage do I use? 5%. TB के लिए क्या था? T20. Lepra के लिए 5 alphabets, 5%. And now you will call this stain as the Phyt Foraco stain. So we always study that if you have used 5% H2SO4, you will rename it into Phyt Foraco stain. Same Zn stain ही है, but because I used 5%, it is Phyt Foraco stain. Phyt 5. Simple. Okay, now like TB had a grading, there is a grading that you have for the lepra bacillus also, Mycobacterium lepra. Because remember I was telling you that 4 plus 6 plus is there. You must be wondering what is 4 plus 6 plus. So that is what is it. So you will find out that what have they done. If you see 1 to 10 bacilli in every 100 fields, then if you see 1 to 10 bacilli in every 10 fields only, or if you start seeing 1 to 10 bacilli in every field, so pehle to you are decreasing the fields. 100 fields mein dikha, matlab it is just 1 plus. You started seeing it in 10 fields, means it's more, 2 plus. If you started seeing 10 bacilli every field, it's 3 plus. Now field to you have come to every field. Now start increasing the bacilli from 1 to 10, make it 10 to 100, make it 100 to 1000, or make it more than 1000. So repeating, first you will talk about the field, 100 field, 10 field per field. That is 1 to 10, 1 to 10, 1 to 10. After that, in the field only, you're going to move ahead. So that is going to be 10 to 100, 100 to 1000, and more than 1000 per field. So that will make it from 1 plus all the way up till 6 plus. So now you will say that, ma'am, how many slides you took? You took 6 slides. Remember, from the chin, from the cheek, from the earlobe, from the chin, from the nasal mucosa and the buttock. 6 slides tha. So for example, you have 6 smears like this, and to each one of them, isko humne diya 2 plus, isko 3 plus, isko 4 plus, isko again 1 plus, 3 plus, 2 plus, like this. Every smear was given this grading. Ye wala jo grading kara, for every smear I gave it. Now I'll have to find out the average bacteriological index. So whatever, so you add this 2 plus 3 plus 4 plus 1 plus 3 plus 2 and divide it. So total whatever you've got divided by the number matlab 6 smears. How many number of smears you counted, you divided by that. That is going to give you the bacteriological index. Means you are counting the number of the bacteria, that is bacteriological index. But you're also interested in seeing, are they intact or are they broken bacteria? You are also interested in knowing their morphology. So you want to find out of the total, how many of them are SFGB. SFGB matlab solid fragmented granular bacilli, tuta futa bacilli. Solid fragmented granular bacilli, wo kya percentage hai? Because please remember, for the prognosis, morphological index is said to be much better than bacteriological index. So bacteriological index just tells you total number kitna hai. Morphological index tells you the morphology, fragmentation, SFGB, and that is a better prognostic factor. Tell me, do you have any culture? No. The first question I told you, this is non-cultivable. Why is it non-cultivable? Because this is an obligate intracellular organism. So what you will be needing for this is the 9-banded armadillo. You can see that this is a beautiful picture of this cute little armadillo that we have over here. It is grown in the 9-banded armadillo. Actually, if you count, it has 9 bands. So it is grown in the 9-banded armadillo, or it can also be grown in the footpad of mice. It can also be grown in the footpad of mice, and that is what you have to know. If you do a serology, yes, you can find antibody against PGL1. What is PGL? Phenolic glycolipid. PGL is phenolic glycolipid. If possible, you can do serology against that as well. Now, because I'm telling you everything about it, I'll tell you the little bit of medicine of Lepramin test also. I hope you remember. Lepramin test is also a type 4 hypersensitivity, means it is a cell-mediated immunity. And how much of Lepramin antigen here also, जैसे Montus में you were using 0.1 ml of TB, here you're using 0.1 ml of Lepramin antigen. Montus also you were doing intradermal injection, here also you're doing intradermal injection. But the interpretation in Montus was just single time interpretation after 48 to 72 hours. Here there is an early reaction and here there is a late reaction. So alphabetical. Fernandez and Mitsuda. Fernandez, F comes pehle, so that is the early reaction. M comes later, so that is the later reaction. Fernandez and Mitsuda, early and late. One is 2 days, one is 3 weeks. Remember, how early, how late? 2 days, 3 weeks. F pehle, M बाद में, 2 पहले, 3 बाद में, days पहले, weeks बाद में, right? So Fernandez, Mitsuda, after 2 days or 3 weeks, if you find a more than 10 mm induration, for Fernandez, you are going to measure the induration, whereas for Mitsuda, again, M and N alphabets come almost together, so you're going to measure the nodule size. So for Fernandez, you're going to measure the induration more than 10 mm, and for Mitsuda, you're going to measure more than 5 mm of a nodule formation. That is what is Lepramin test positive. Now, you started giving treatment to the patient. I think this much of information everyone knows now, that we have what is the treatment that we give. If I ask you what is the treatment, there is a DCR regime that we follow now. There is a dapsone, then there is a, just for your quick revision, clofazimine, rifampicin. This is the treatment that we give, the DCR treatment for 6 to 12 months. You started giving the treatment, or maybe you did not give the treatment. So what are the reactions that can happen? So please remember, there is something called type 1 lepra reaction, type 2 lepra reaction. Type 1 can occur before or after treatment. 1 wala can occur even before or after treatment, but type 2 is always seen after treatment, not before treatment. तो अब इसमें ना best way to analyze is type 2 में write the 2 and everything is gonna come out 2. So ये क्या है? This is treatment related. It is related to the T for related to the treatment. Okay, now you'll first ask me that what hypersensitivities are they? So type 1 is gonna be type 4 hypersensitivity reaction. Type 2 is type 3 hypersensitivity reaction. So there is a rule of 5 that we follow over here. If you ever get confused that type 1 lepra reaction versus type 2 lepra reaction क्या था, total 5 बनाना है. So over here if you want to make a 5, you will do 1 plus 4 is 5, and over here if you want to do, you will do 2 plus 3 is 5. So this becomes hypersensitivity 4, this becomes hypersensitivity 3. So rule of lepra, L-E-P-R-A, lepra में सब कुछ 5 है. How much of H2SO4 were you using? 5%. What is the rule that you follow? 5 का rule. So hypersensitivity याद हो गया. Now, let me tell you that type 1 could happen before treatment also, after treatment also. Before treatment, the patient is going to deteriorate only. Bina treatment ke, the patient is not going to be fine. So, patient is going to be deteriorating, means the patient is going to be downgrading. Downgrading means he's going towards the lower immunity. Lower immunity means he's going towards the LL wala pole. And after treatment, patient is going to be better. Might become better, reversal or upgrading. This means he's going towards the better wala pole, the TT wala pole. So simple, this could occur before or after treatment. Before treatment, he'll deteriorate, downgrading to LL. After treatment, he might get better towards TT. But type 2 is definitely gonna occur after treatment. So this is seen in all those borderline BLLL cases. And what exactly will you see after treatment? That is known as erythema nodosum leprosum, written as ENL. Type 2 is known as ENL, and now I'll tell you all the T's that are affiliated with it. So what are the T's that are affiliated with it? Firstly, when I said erythema nodosum leprosum, you will learn that they are T for tender nodules. Why are they tender? Because of release of T for TNF alpha. What is the hypersensitivity response? It is the TH2, again T2, not TH1, TH2 response. And what is the treatment? It is going to be T for thalidomide. So remember, when you're talking about, repeating, when you're talking about type 2 lepra reaction, this is treatment associated. This is treatment associated. It is going to show you tender nodules, that is known as erythema nodosum leprosum. It is going to show you that tenderness because of TNF alpha. It is going to be associated with TH2, and it is going to be treated with thalidomide. That is type 2 lepra reaction. Whereas what do you see in type 1? Type 1 may either there is some inflammation, swelling, or there is an ulnar nerve involvement. Ulnar nerve involvement may, if you exert pressure on the ulnar nerve, I hope you remember there is a tinel sign. ये T उधर मत डाल देना, ठीक है? Ulnar nerve वाला जो sign है, that is if you exert pressure, you will see tingling sensation. That tinel sign is seen with type 1. That's a very famous dermatology question that you get. And finally, ये TH2 नहीं होगा. This will be TH1, and you will not be treating it with any thalidomide. You will be treating it with steroids. So simple way of learning was everything to do with type 2 had to be 2, 2, 2, 2. ठीक है? Simple. Type 2, repeating, type 2, treatment associated tender nodules called erythema nodosum leprosum because of TNF alpha, TH2 response, thalidomide treatment. Now, finishing off the entire Mycobacterium chapter means you're ready for quick 1 or 2 questions. Can you tell me all of the following are correct matches except Mycobacterium cansaci is a photochromogen. Yes, photochromogen के लिए mask था, so cansaci is definitely photochromogen. Batis bacillus was Mycobacterium avium complex का intracellular non-photochromogen, correct. Sulgi was photochromogen, God save us from the dark, correct. Xenopy was photochromogen, nahi, photochromogen was mass. Xenopy was that non-photochromogen. Xenopy was non-photo miscellaneous category, so that is the answer over here. Coming on to the next. Mycobacterium TB. Now, this is a question that I have got in the end because no one really studies it anywhere, but you will just find this one question that has come in your previous year, so I want to discuss this. How do you differentiate Mycobacterium TB from all the other mycobacteria? Answer to this question is going to be niacin, and that's the last part that I wish to discuss with all of you. So please note there is a mnemonic called NNC, and I'll take you back to one table which is very, very important for all of you. Have a look at this table. What is NNC? So N for, you can see definitely niacin that has been put up over here. So here you go. So to differentiate Mycobacterium TB from Mycobacterium bovis or others, you have to follow something called NNC. This I'm only telling for INI-CET aspirant. Remember, TB is positive for all. Means it is going to show you the niacin test, the nitrate reduction and the catalase positivity, whereas bovis is going to show you all negative. Also, I have told you TB strictly utilizes oxygen. So this means bovis is going to utilize less of oxygen, microaerophilic. Also, I have told you TB has a rough, buff and tough colony. Yes, Mycobacterium bovis is going to be opposite. Rough ka opposite, smooth. Buff is cream, na? So this is going to be white. And tough. Tough basically means that it's very difficult to remove. On the other hand, bovis is going to break easily. So repeating, bovis is the opposite of rough, buff, tough. TB was rough, buff, tough. Bovis is going to be smooth, white and will break easily. So remember, TB is NNC positive, niacin, nitrate reduction as well as catalase. Bovis is not. So that takes you to this question and you can easily answer this now. I hope you're not going to have a problem over here. So coming back, Mycobacterium TB is differentiated from other mycobacteria by which of the following cells or test? So not pass 10, pass is nowhere in the rule. AFB is though positive in both, so niacin is going to help you. Actually, there's one more question that you have, a clinical question. A 66-year-old man underwent a hip replacement, तो कोई surgery हुआ है following a degenerative joint disease, and 5 days later he's come with fever and pain with tenderness and swelling in the hip joint. Hip joint shows accumulation of fluid which is when aspirated sent for microbiology culture, तो कोई तो abscess बना है, right? And that has shown positive growth in 4 days. And the organism is acid fast. Acid fast matlab they are talking about the Mycobacterium species, and 4 days, less than 7 days means it's a kind of a rapid grower. Which of these is the rapid grower? I can see this Chalu person over here, Chelone. So Chelone is going to be the rapid grower, right? So that is going to be the answer over here. I hope everyone's okay with this question, because with this we have wrapped up our entire discussion of